welcome to this IChemE Global Awards 2021 webinar. My name is Matt Stalker and I'm your host for today's session. The IChemE Global Awards celebrate chemical, process and biochemical engineering excellence and are widely considered as the world's most prestigious chemical engineering awards. Today we'll be revealing the winner of the IChemE Pharma Award, sponsored by PM Group. This award recognises the best project, process or product to demonstrate chemical engineering excellence in the pharmaceutical sector. So before we check out the contenders, let's hear from our sponsors. Please welcome PM Group's Regional Development Director, Peter Farrelly, and Director of Architecture, Jordan Kemp.
I hope everyone can hear me okay. Good afternoon, everyone, and welcome to this awards event. We are especially pleased to sponsor the Pharma Award as part of the IChemE Global Awards 2021. The last 18 months have certainly shown us the importance of the pharma sector in all of our lives and the key role that the chemical engineering professionals have played in the development and rollout of vaccines across the world. In PM Group, we have a long history of designing and delivering vaccine manufacturing facilities, and our chemical engineers have played a central part in all of those projects. And these projects are complex, with innovation and cutting-edge engineering at their core. In the pharma sector, we've now moved well beyond the simple concept of having automated factories, and now the concept of smart manufacturing is an integral part of our facility design. And this is something that has to be designed into the buildings and equipment right from the start. As an international project delivery specialist, we're very comfortable with complexity and innovation. In fact, these are things that are at the heart of all of our projects. For almost 50 years, we've been helping our multinational clients to design and deliver their latest facility developments across the world. The skills and expertise to do that successfully have to be nurtured and built over many years. And for us, that starts with our graduate training scheme, a scheme that's recognised and supported by IChemE. And then that expertise is built up over time through continuous professional development activities. The IChemE Global Awards are helping to inspire greater investment in this kind of expertise, leading to greater innovation and, we hope, a more sustainable future for all of us by the use of advanced smart manufacturing technologies. I'm pleased to now hand over to my colleague, Jordan Kemp, our Director of Architecture, who will talk to you more about smart manufacturing and how it's helping our clients and industry alike. And I just wanted to finish off by saying best of luck to all the finalists. Thank you.
Hi, everybody. I'm pleased to present today. Just a brief presentation just to socialize some of the key concepts that we as a company has been looking at for our clients, as Peter highlighted there. So smart manufacturing and Industry 4 is no new concept. It's been something that's been out in the industry for probably up to 10 years now and well established in the automotive and the aerospace manufacturing industry. Some of the principles and the key technology pillars is well understood and it's identified. So, but now a lot of this is making its way into the pharmaceutical industry and has a direct impact on how we deliver our projects and how we design our facilities to our clients. And in the end of the day, what is it trying to achieve for us? It's aiming to have facilities that is cognitive and drive towards a digital transformation. A factory that not only understands the product requirements and its current status, but is also able to predict and prevent failure to avoid shutdowns. So how do we achieve this? A huge level of automation. So in a lot of our projects, this element is driving a lot of change, connecting up the data between systems, making things talk to one another is some of the biggest challenges within the projects, because all the data now needs to be analyzed. We need to make sense of it, and then that is driving the decisions that the facility needs to take. So if we have cobots in the facility, the cobots needs to be told to go and fetch something in the warehouse, take it to the kitting area, move through the facility. It needs to send the right cobot to the right place within the facility. So all of this data integration is something that starts absolutely at the kickoff of every project, and a lot of our clients is grappling with this integration right now. And on a couple of our projects now, we have successfully integrated this, and starting all the way from just simple things like setting up tagging conventions correctly, because you can't get to the end of the project and your things are not talking to one another and they're not integrated. So just to look at a brief example, like we're currently working with a client in vaccine that's asked us to standardize a vaccine manufacturing facility and copy that across the world. So some of the challenges was on like smart buildings is the environmental control, because each building is in a different place in the world. So the envelope to respond to its location and also to look at not overheating, a simple challenge. The industry is well versed on this type of systems, but to make sure that it can adapt to wherever this facility is located. Also, when we looked at robotics, there's not just one robot that can do everything. There is, in this facility, there's up to 5 different types of robots that can actually facilitate lifting and moving, or lifting up one high. It's 2 different types. It's not just one type that can do that. Some of the cobots that's changing gowning, finishing garments, and simply just moving from one area of the facility to another with empty drums. So integrating this has been a challenge, and the industry is still to a certain degree grappling with how to do this. Storage and logistics, it's always an aim just to have just in time and not be building large expensive warehouse areas. But if your supply chain is not linked to this building and your supply chain is not delivering your equipment to the facility in time and the way you want it, then that needs to be integrated, and it is a potential issue. A lot of these items, virtual reality, cloud-based analytics, like creating data centers, and then most importantly, what we all of you are trying to drive it is real-time release, integrating the QC systems or automating the QC systems. This facility, we now have a system that does environmental testing, where the cobot grabs the environmental plate and moves it to a machine that actually tests this automatically, and so it no longer travels to the QC facility for a human intervention and human testing. The digital twin, touching again on the data analysis and starting at the kickoff of a project to make sure that the data is dealt with and set up correctly, so that the digital twin in the end of the project is actually doing what it's supposed to doing, and it can react to maintenance requests or actually bring up the system as requested, because integrating it at a later stage is a mammoth task going forward. And what is this all driving to? We're all aware of our climate challenges, and in PM Group, we strive to now drive all of our buildings to zero carbon facilities, where how smarter the building is and how better the building can react to its environment, actually assisting this agenda. So it's something that is complete part of this cognitive building design. Another element that is driving this aspiration is also standardization, and it's also embracing this as a part of every facility design, because we can't have all the aspects anymore bespoke, because the integration of that bespoke piece is quite considerable and takes a long time to integrate with new systems. So the more we can standardize components and set up the interfaces, the faster this facility design can be achieved. The next step is also the team structures. So how does the team structure now change? Is this third person, like for 20 years of my career, I've been looking at the process architect and the process engineer being joined at the hip for designing facilities. But now we have smart manufacturing specialists that needs to help and coordinate the integration of systems within the facility. And that person starts at the kickoff. It doesn't start halfway through the process, it's too late. And then the whole digital journey is to understand the digital journey, and that it's a process that needs to happen. And a lot of it is not a single step. It's starting to set up on that journey, recognizing that we need to digitize what we have, our records, and driving towards a fully digital transformation. There's a lot of hurdles on this because some of the technology simply isn't available. And the question is not when it will come, but we need to enable these technologies because it is coming. So understanding that, setting our data up in such a way in our tagging conventions so that it can integrate with whatever is coming down the line. Also people, like getting people to understand the benefits of this, and to help them understand that the benefits to the business, and that resistance to some of this change could lead to failure in the future. So what is the future? I know for a fact that I'm not trying to design humans out of facilities, that's for sure. But I know change is coming, and in PM Group, we want to be part of that. And the whole connectivity between what's happening within the world, we need to make sure that our facility designs is embracing that. So we know we cannot consider smart manufacturing as something that can be bolted onto facility design. There's a lot of planning and integration required upfront to be able to enable a better connected full life cycle facility design and integration. So I thank you for your time, and hopefully this was a bit of a thought-provoking presentation, and good luck to all the finalists going forward. Thank you, Peter.
Thank you, Peter and Jordan, and thank you to PM Group for its support of the IChemE Global Awards. Now then, our awards judging panel has been hard at work in recent weeks to review and rank all of the entries, and here are our finalists. We have GSK Research and Development, GSK again, Micropore Technologies, a joint entry from North Carolina State University and Photocide Protection, and finally another joint entry, this one from the University of Sheffield, Perceptive Engineering and Siemens. All our finalists have been invited to join us today to answer your questions and to tell us more about their work. You can get in touch throughout today's webinar using the questions box on screen. So let's get started with our first finalist representing GSK R&D. It's Anthony James.
Right. Hello, thank you very much for the opportunity to be here. I'm Tony James, and I'm representing the team that's done fantastic work on making early phase medicine manufacture safer. So pharmaceutical manufacturing comes in two parts, just a little bit of preamble. So for a couple of decades, my world was primary manufacturing, and John's talk, which is up next, is in this world of reactive chemistry, solvent use. We're making the active ingredient, and pretty much everything in this world is inerted inside the equipment. The last decade or so, my world's been secondary manufacturing, which is taking that API, formulating it with additional materials to make the tablet or whatever it is that we take as patients. And in this world, very few unit operations are inerted. Most things are done in air. And that knowledge that things are done in air is important to make the rest of this work make sense. So we set out to solve a couple of problems. The first is to understand what process safety information we really need to know, what's our absolute minimum needs, and what controls must we have in place to safely manufacture in the early phase environment. And this term early phase means quite early in research and development. We're just starting to put the material into people and into patients for the first time. And the second part of this work was to work out what do we do if this information's unknown and we've got no real way of knowing it. So when I was first out of university, life was much easier. If I need to know the property of a material, I'd look in Perry's. If that didn't work, I'd wander down to the technical library. And nowadays it's so much better. It's information just a few clicks away, except in early phase medicine manufacture, because it's a very data poor environment. The things we're working on have only just come into existence and there isn't very much of it. And that's our real difficulty. There's very little material, at most probably 10 kilograms in existence, often less. And it's very hard to spare the kilogram or so that it takes to get full fire and explosion data, because really that material needs to be inside patients, and it's quite rare to be able to spare the material. And internally, that means we have to have very burdensome safety measures in our secondary pilot plants to keep our people safe. Other people take a different approach, and sometimes, unfortunately, there are accidents. And we set about the two arms of this work to make ourselves safer. And the first arm of the project is, what do we need to know? So do we understand what can go wrong, what control measures we need, and do we know those control measures are working? Apologies, the next slide's a bit complicated, but we have about 4 property-dependent ways of igniting materials. We can either have a fire, we can have a dust explosion, not an ignition, but we can have a thermal decomposition, which is nearly as bad. And there are some things we need to know to prevent those bad events. And in some cases, there's more stuff we need to know to mitigate those events should they occur. Maximum recommended processing temperature only takes a few milligrams to get, so we get that, and we get the information to mitigate. As I said just now, the rest of those takes about a kilogram of material. Often we simply don't have a kilogram of material. So the team's done a lot of work going through unit operation by unit operation in the secondary pilot plant world to understand whether these various sources of ignition are credible, whether we absolutely need to know those properties, the controls we have to have in place. And as well as that, it's involved a lot of going out and really looking at and understanding the equipment, doing literature reviews, doing calculations of charge generation potential, kinetic energy, sometimes angular momentum, all to really understand this. And what we end up with each unit operation is a several-page document, and some unit operations come out as very benign. There isn't much that we need to know. We have to comply with the ATEX certificate, but things are relatively benign as long as we have our controls in place. And other unit operations are very different. So I've highlighted co-milling here, but there are others where there are properties where we absolutely have to know. We have to know the minimum ignition energy and thermal stability to co-mill. And as we've looked at this and gone into it and thought about it, there's a few occasions where we've added restrictions beyond the ATEX certificate, and we've really had to understand what are our critical controls that have to be in place and functioning to keep us safe. And what we end up with is this rather nice summary chart, which sits in the plant introduction risk assessment process and is delivering a huge amount of value. So you can see there's lots of places where actually it doesn't matter too much if we don't know properties. There are more places in red where we absolutely have to know the properties. And inevitably with these things, there's a grey area, in this case masquerading as a yellow area, where things are a bit more nuanced. But for all that to be effective, the controls have to work. So we went into this, and particularly I was thinking that our barriers were really strong, but the more we looked at our barriers, some of them weren't quite as strong as I had thought they were. And I want to congratulate the pilot plant team on the integrity, determination, and technical excellence they've put in to making these barriers more robust and building better systems, not necessarily more complicated systems, just better and more robust systems. I particularly like to call out the new Thera process, which is actually less bureaucratic than the old system and much stronger, and we're starting to build in barrier effectiveness even before we bought bits of equipment really early on in the design, marrying together operational and process safety, which can be difficult at times. Although it's not on this slide, I also want to highlight the work of Sydney Lang and her fantastic resilient system for pre-start earthing checks. So that was all about understanding what we need to know. And the second part of the work is, well, what do we do if we don't know it? If the first part of the work says we have to know minimum ignition energy or we have to know dust explosion properties, well, what if we don't know them? And what if we don't have the material to go away and find that out? And this work's been recently published, and there's the link if you want to see it. Although it's technically interesting, what this work is about is making sure people stay safe. So what we were after was a methodology to predict the properties, particularly to highlight materials of concern. And for us, material of concern is one which is particularly susceptible to ignition, has dust explosion properties beyond those we can normally mitigate, or can catch fire at a much lower temperature than we would normally expect. And our success criteria were we wanted to use less than a gram of material to do this. Although we're a large pharmaceutical company, we didn't want to spend any money. We could accept a level of false positives. What we really didn't want were any false negatives. So going on to minimum ignition energy, there's an existing literature model which we can solve for many of our compounds. We can have a typical range of densities, typical range of specific heats, and that equation's solvable. In the intervening time, we've known a little more about spark generation efficiency. We had to put in a correction for that, but it comes out about there, 9 to 26 millijoules, typical minimum ignition energy. But over the course of time, GSK's done a lot of fire and explosion testing. We've got a big back catalogue of results, and we can compare this prediction against the historic data. And what you can see is we're in the right ballpark, but that Calcut model isn't giving us the range. But start to look at the molecules behind those bars. Something interesting emerges. On one side, we're seeing really stable molecules held together very tight crystal lattice. And on the other side, there are molecules with obvious structural weaknesses, very large decomposition exotherms, and or really weak crystal structures. So what we've done is taken that Calcut equation as our base and as our model. So the two comes from spark generation efficiency. Then we have correction of factors for thermal stability, intermolecular stability, and the stability of the molecule itself. And to run the model, we need some good chemists to look at how molecules cleave. Unfortunately, we have excellent chemists, and we need to do 2 experiments. Those 2 experiments only need 50 milligrams of material. In the future, I think there's space for another factor around the shape and flow properties, agglomeration and dispersion of materials, but that's more important for dust explosions. So having a look at how we're doing, see at the top, compounds A and B, we've correctly predicted materials of concern. Next one down, there are 2 compounds, actually now 3 compounds, where we predicted materials of concern. We've been overly conservative, but we haven't been wrong by a huge amount. Then there's a big bunch of them where we said it's not material of concern, and we've been pretty accurate. There's a couple at the bottom where we said it's not material of concern, and we've been excessively conservative. But overall, MIE, minimum ignition energy, is going well. On dust explosions characteristics, it follows on from the MIE work, same sort of hypothesis, but the dust explosion property is actually dominated by the most readily flammable portion of the molecule. Sometimes it's the whole molecule, but more often it's a flammable fragment that's cleaved from its parent molecule. The Pmax we get by a thermodynamic combustion of the flammable parts. That's undergraduate thermodynamics, which is just about the only thermodynamics I'm still capable of doing. And it's based on the cleaved flammable fragments, where there are fragments, or sometimes we do the whole molecule as a safeguard. Heat of combustion comes from Trotter or from published data if the fragments are a really common molecule. KST will come on to the next slide. So KST or ST class, that comes from the assumed Arrhenius type relationship where you have an expansion factor, because by and large there are more gas moles post-combustion than there were pre-combustion. Free exponential factor is based on minimum ignition energy and the percentage of the molecule that's combusting, minus E over R is related to heat of combustion, and T's the adiabatic flame temperature from the combustion calculation. And how this one's going, we set a bar a little bit below our true material of concern level to allow for the fact that the model's not perfect. No model's perfect, and this one's no exception. And see, at the top, we've correctly flagged again a couple of materials of concern. We've had a couple of false positives, and one of those we were really a long way out. A bunch of 4, again, reasonably accurate, and a couple at the bottom where we were right, they're not materials of concern, but again, we were a long way out. And the reason we think we get so many that are a long way out is the model assumes the powder disperses perfectly, and we've got some evidence that not all of our powders do disperse perfectly, and we're looking to see if we can somehow build that into the module. Whether we'll succeed or not, I don't know yet. Minimum layer ignition temperatures follow the same theory. In this case, it's a flow chart, but it's the same theory. It's how the molecule cleaves. Is it cleaving something that has particularly low auto-ignition temperature? Does it have a sufficiently big decomposition exotherm that it could self-ignite or go a long way towards self-igniting? If it does, it have any accelerants based in the chemical structure? If not, it's probably quite a benign T class, and then maybe less benign, or in the extreme case, we really have to go away and test. So in conclusion, for the first part of the project, we now know what we need to know for each unit operation, and that's allowing us much faster, much safer, and much more consistent plant entry into the early phase pilot plants. Our controls are more robust and our systems are more resilient. For the second project, if we have the material, we should always test. The prediction is no substitute for testing if there's material available, but if there isn't, we're finding that we are predicting materials of concern, with the caveat that our sample size is a bit small. We're meeting our success criteria. We think there's a bit more to do on dispersion. The time I wrote this, 22 compounds up to 24 compounds now we've used this on. I'm conscious that I'm running out of time overstaying my welcome. I had a fantastic team. I'd just like to call out in particular Rebecca, Bea, Eddie and Sydney, because these are young engineers coming up to chartership, or in Eddie's case, engineering apprentice. I just want to say the future of engineering is in good hands. Thank you.
Okay, thank you very much, Anthony. So it's now time for questions. If you would like to get in touch and haven't already done so, please use the questions box. We do have our first question. How did you discover this gap in your safety processes? Was there a near miss, or was it caught in some internal safety procedure audit?
Thank you. A mixture of two things. So we were aware of it, and it was kind of bubbling along in the background, and we were working on it. And like I think many people do, we look at accidents that happen throughout industry and in other companies, and we saw one where there was a powder fire, and the powder fire was ignited by a lack of control of static. But one thing that came out of the investigation was the material that caught fire had an unusually low minimum ignition energy, which the company hadn't known at the time. It came out of the investigation. So that prompted us to think, well, why didn't they know? And why didn't they know that they needed to know, if that makes sense? And we reflected on that internally, and that's really part of the genesis. We gave extra momentum to work we were already starting to do. And hopefully that answers your question.
Great, yes, thank you. We're going to move on. So thank you very much for getting things started, Anthony. It's now time to move on to our second finalist. Once again, it's GSK. And for this entry, we have a pre-recorded presentation.
Hello, my name is John Lim. It's an honor to represent the GSK team as I'll be talking today about our work in a safe and effective deactivation of rainy cobalt using aqueous sodium nitrate. Now, the reaction in question is shown here on the title slide. We're trying to reduce benzonitrile 1 to the benzylamine 2. Quite a bit of screening went into identifying rainy cobalt as the ideal catalyst for this transformation. Now, because 2 is taken through 3 more stages of chemistry before the API step, it's imperative that we have a clean reaction profile in this reduction, which is what rainy cobalt gives us. However, with any heterogeneous catalytic reaction, there's a filtration involved, and thus there's an inherent risk if that catalyst bed were to run dry. So this is what happens when rainy cobalt gets dry. So what I'm doing here is something you should never do, and filter the catalyst to dryness. The image on the left is taken with a thermal imaging camera, the image on the right, which is a standard video camera. And as you can see, upon reaching dryness, the catalyst bed reaches temperatures in excess of 350 degrees, which is the detection limit of the camera, and thus the actual temperature is probably much greater than that. So the pros of using rainy cobalt is that it gives us a very clean reaction profile using an earth-abundant metal. The cons is that because we're loading 10 to 28% catalyst, that's a lot of catalyst that we have to handle and dispose of later. Now, we had a pilot plant campaign planned at our Cork pilot plant where we anticipated making about 100 kilograms of API. Now, you can imagine if we're charging 10 to 28% catalyst, that's 20 kilograms of catalyst or so. Well, you don't want that much catalyst reaching temperatures in excess of 350 degrees in just a few seconds. And thus the Cork pilot plant requested that we come up with a way to effectively deactivate the rainy cobalt so that it was safe to handle even when dry. So in other words, we couldn't just drum up the stuff underwater, we actually had to deactivate it. Now, a common way to deactivate rainy metals is using bleach. And so what I'm doing here in this video is I have a little bit of rainy cobalt in the graduated cylinder. I'm adding typical household bleach solution. Now, as the video comes into focus, you'll notice some vigorous bubbling happening. Now, this is concerning. You don't want this to be happening in your reactor. Now, it's not too dangerous here in the fume hood, but you don't want all this off-gassing to happen when you're trying to quench the catalyst in a reactor. What's also very concerning is that the temperature of the slurry was about 20 degrees at the start before the addition of the bleach. Upon adding the bleach, you notice very rapid exotherm in excess of 20 degrees. Now, keep in mind, this is not really a controlled reactor. This is open to air, so there's probably some heat loss to the environment, and so that 20-degree exotherm is probably not indicative of the actual heat that's generated. There's probably more heat than that. And thus, just qualitatively, we can see that the bleach quench is not safe. It's exothermic, and we see a lot of gas evolution upon quenching. Now, I should say, though, that the bleach quench does work. The catalyst, after it's been treated with bleach, is deactivated. However, there's some hazards that are clearly evident just upon qualitative observation, but we wanted a more quantitative evaluation of the hazards of using bleach to quench rainy cobalt. So in order to quantitatively assess the hazards of using bleach to quench rainy cobalt, what we did is we took 35 grams of rainy cobalt from our supplier. We put it into a par vessel and hydrogenated with about 4 or 5 bar of hydrogen to mimic reaction conditions. We then took that activated catalyst and we measured the heat and off-gassing when quenching with 20 volumes of 5% bleach in what's called our RC1, or reaction calorimeter. The green trace represents the addition of the bleach. The red trace represents the heat output. The blue trace represents the off-gassing. And what you notice is that once we start adding the bleach, there's an immediate exotherm. In fact, there's so much heat we actually have to wait for the reaction to cool down before we add the rest. Now, once we resume the bleach addition, we notice another exotherm and very significant off-gassing here, and the gas we later identified as oxygen. So at the end of the reaction, we did the calculations, and we found that the total heat output was about 3,800 kilojoules per kilogram catalyst, which equated to a 53-degree adiabatic temperature rise. Now, a typical plant reactor can handle about a 20-degree adiabatic temperature rise, so this is well in excess of that. What was also very concerning is the gas evolution, 91 liters per kilogram of catalyst. And so once again, if you're charging 20 kilograms of catalyst, you do not want to be producing about 1,800 liters of oxygen when you're trying to do the quench. Now, the pilot plant staff, upon seeing this data, were understandably alarmed, and thus they would not run the rainy cobalt process as we designed it using the bleach quench. And at this point in the project, there were a lot of meetings happening, a lot of brainstorming, a lot of ideas that didn't actually work. And so this put the project timelines in jeopardy. So at this point, I did what a lot of people do during times of stress. I went to Google. Here is a screenshot of the Google search I did that day, deactivation of rainy metals, which brought back this patent from 1957 from the DuPont company. And here the authors describe a deactivation of rainy nickel using aqueous sodium nitrate or nitrite. They said that the aqueous sodium nitrate was a little bit more effective. Now, it's one of the shortest patents I've ever read, and I'm glad I read to the end, because at the end the authors mentioned that rainy cobalt could also be deactivated in the same manner. So we decided to try it out for ourselves. So we did the same reaction as before. We took 35 grams of rainy cobalt. We activated it under 4 or 5 bar of hydrogen, but this time we quenched with 20 volumes of 5% sodium nitrate in an RC1 where we measured the heat and off-gassing. So I showed the data for the sodium nitrate quench on the left and the data for the bleach quench on the right. So once we begin our addition of sodium nitrate, we do see a spike in heat, as you see here in the red curve, but the total heat is actually measured as the area underneath the red curve, and so the area underneath the red curve on the left is much less than the area underneath the red curve on the right. Now when we compare the gas evolution, which is these the blue traces here, when we start adding sodium nitrate, we do see some spiking here, but you notice the difference in scale of the y-axis, which measures the gas flow. For the sodium nitrate quench, we notice that it's nearly 50-fold less than the bleach quench. So this spiking that we see here is really not much more than baseline noise. There's very little gas evolution with the sodium nitrate quench. So when we compare the numbers for each quench, we see that the sodium nitrate quench produces about 4-fold less heat per kilogram of catalyst, which equates to an over 4-fold reduction in the total adiabatic temperature rise, and this value of 12 degrees is well within the safety limits. We also see that the sodium nitrate quench produces very little gas and no oxygen. And so what we think is happening in both quenches is that we're oxidizing the surface-bound metal hydrides to deactivate the catalyst. But with bleach, what happens is that we're producing oxygen, whereas with sodium nitrate, we just produce some nitrogen, which we know is inert. We also see how much more efficient the sodium nitrate quench is. We see that 2 moles of sodium nitrate can quench 10 moles of the cobalt hydride, and that whereas with the bleach quench, it takes 3 moles of sodium hypochlorite or bleach to quench 2 moles of cobalt hydride. Now, this is a hypothesis of ours, so in order to test it, our process safety engineers perform what's called a cheetah calculation. And so cheetah is just a standard process safety calculation where they can estimate the heat output based on published data. And so when we do the calculation based on these proposed balanced chemical equations, the expected heat output for each quench is very close to the observed value. The numbers are all well and good, but does the sodium nitrate quench actually work? Does it actually deactivate the catalyst? So what I've done here is I've taken some activated rainy cobalt, I've slurried it up in some sodium nitrate solution, and I'm going to filter to dryness again. Now, a lot of presenters will save the exciting video for the end, the one with the big explosion. I'm going to do the exact opposite of that and show you the most boring video here at the end of the presentation. As I filter to dryness, absolutely nothing happens. So I measured the temperature of the catalyst bed for 45 minutes. I won't make you sit through all 45 minutes, but in all that time, the temperature of the catalyst bed never gets above 20 degrees. The catalyst has been completely deactivated, stable to open air even when dry. And so this material is sitting in my fume hood as we speak, completely happy. No self-igniting behavior is ever observed. And thus, upon seeing the data, the rainy cobalt process was approved for our pilot plant campaign. So if you care to read about this further, we published an OPRD not too long ago. And finally, I just want to acknowledge the whole team, the process chemists in chemical development, our process safety colleagues who performed the calorimetry experiments and the cheetah calculations, and our colleagues at the Cork pilot plant. I'd also like to thank you all for your kind attention, and IChemE for their gracious consideration of our work for this award.
Okay, that was a pre-recorded presentation, so no Q&A. Instead, we'll move swiftly on to our third finalist of the day. It's Micropore Technologies. Please welcome Sam Trotter.
Hello, thank you very much. Just share the screen there. I'm not sure which screen it's shared. I don't know if anybody can help me out there.
Yeah, that's perfect, Sam.
I'll share the right one. Wonderful. Cool. Well, yeah, thank you very much. Yeah, so my name's Sam from Micropore Technologies, and today I'm going to talk to you about our versatile lipid nanoparticle manufacturing platform. First, we'll start off just a quick overview of the structure of a lipid nanoparticle. So what you can see there is a kind of artist's impression of what a liposome or a lipid nanoparticle may look like if you get this sort of view of one. And what these particles essentially are is a lipid layer surrounding a core where you've trapped some material inside. These can be lots of different things applicable to very different types of drug delivery. The way that most people will probably hear of these or be aware of them at the moment is their use in vaccine delivery, wherein some mRNA is entrapped within the lipid nanoparticle itself there. So by using different combinations of lipids and different materials, you can create these particles for lots of different applications. My colleague likes to say these have been an overnight success story, about 20 years in the making, things that people have been aware of for a very long amount of time, but have only really recently come into the public eye in terms of their huge potential for being able to deliver things inside of cells. The traditional way for manufacturing these materials is essentially mixing together two solvents. This is what's commonly referred to as the ethanol or solvent injection method, whereby we have our lipid mixture in an organic solvent such as ethanol, and we have an aqueous buffer solution. So if we're making these mRNA-loaded lipid nanoparticles for vaccine delivery, and then we're mixing those two together in a mixing process. And as that happens, our organic solvent transfers into the aqueous phase. That results in our lipids precipitating out. And as a result of that precipitation, they can entrap some of the aqueous material. Even in the case of mRNA, the opposite charges on the mRNA and on the lipids cause those to essentially be drawn together and form those lipid nanoparticles around the mRNA. And that is essentially the process that is being used today for the creation of the vaccines that I've certainly been injected with, and obviously countless others as well. Now, these formed vesicles, depending on that mixing process, can have various different sizes, structures, and size distributions depending on how that mixing process is controlled. For instance, if your mixing hasn't been controlled properly, you can end up with what's called a multilamellar vesicle. So rather than just one nice individual wall, as we're seeing in that diagram there, you can have walls within walls within walls, which of course, when you're trying to release something in a predictable way, is less than ideal. You can also have wide size distributions. When you're injecting something, you want to be able to predict where it's going to go and how it's going to behave. Having those uniform sizes is very important. Additionally, a lot of these processes are terminally sterilized via sterile filtration. So trying to pass materials in the nanoscale with wide size distributions through a sterile filter can result in significant losses if you have quite a wide size distribution. And that's the issue with some of them being too large, as you have very heavy losses. And when you've just spent millions of pounds synthesizing your RNA, the last thing you want to do is lose it on a filter right at the end. So the mixing process is absolutely the key to successfully forming these particles. A lot of the current processes based on microfluidic mixers and impinging jets, my crude interpretation of one on the right there, essentially we're just blasting two jets together, and then the resulting solution is where that solvent is diffusing into the aqueous phase and we're forming those particles. Some of these systems can have quite limited throughputs, especially in the case of microfluidic systems, good on the small scale, not so much on the large scale. So that gets into difficulties if you're trying to make lipid nanoparticles for a few million people, say. Some of these systems can also take very high pressures as well, and that can make process controls difficult. You're trying to constantly manage these two high pressure pumps that are blasting against each other, can be difficult for those control procedures. It can also introduce quite high shear as well for something when you're handling mRNA, which is quite a sensitive molecule. Those high shears not necessarily desirable for this sort of process. You can also need to change equipment for different scales. So your small scale, you might be working with microfluidics on your benchtop. Then you have to go to a high pressure impinging jet as you get towards production. And having to do that equipment changeover after you've spent a lot of time developing your process already is not going to make anybody happy. So we saw this as an opportunity to apply our experience with membrane emulsification, essentially forming droplets and emulsions, to apply that membrane technology to forming these lipid nanoparticles. So what we've developed is a platform that's based on Micropore's patented membrane technology. We've developed this using our stainless steel membranes that you can see on the right-hand side of the screen there. These essentially are stainless steel tubes that we have very precisely formed pores in them, controlling the size, the distribution of those pores to a very high degree, as you can see from that micrograph. This gives us a system that's scalable across the 3 devices that you can see on the bottom of the screen there, from 1 milliliter per minute all the way up to 1500 liters per hour, all using this exact same type of membrane, this exact same type of mixing technology. These devices, all 3, designed for very low holdup continuous operation, just as comfortable producing a milliliter as they are producing hundreds of liters. All designed for aseptic production, so even if we weren't producing very narrow size distributions, which we are, you could run these fully aseptically. That of course adds the capabilities to produce larger particles than had previously been possible with these sort of nanomaterials, because you've been limited by what you can put through a sterile filter. All of these have also been designed to be robust, easy to clean, and have minimal consumables to make them usable at all stages of developing these lipid nanoparticle products. How does all of this actually work? Essentially, what you're seeing there is an overview of one of our devices that we call the AXF1. It stands for Aseptic Crossflow, and it's got 1 tube in it, so it's a 1. And what you can see is our continuous phase on the left. That would be our aqueous solution, our aqueous buffer, essentially containing the mRNA. The dispersed phase would be our solvent containing our lipids. As we're passing it through those pores in the membrane, it's being entrained in that continuous phase flow, not wholly unlike a jet in crossflow. Some of you are more familiar with those types of mixing applications. And then we're getting that very controlled micromixing occurring inside the device. These membranes that we're placing inside are essentially a very much scaled up version of similar single pore systems. So if you're familiar with microfluidics and single junction T mixers, imagine 1.7 million of those within a tube that's 6 inches long. And that's what we've created with the AXF1 here. And that gives us this excellent scalability and control that we think is quite unparalleled in other technologies that are available for producing these particles. So we set out to demonstrate that this was suitable for producing the type of particles that are required for vaccines, given that that was the immediate need, is how can we help people to develop these vaccines from those small scale benchtop requirements to those full large scale production needs as well. So we started out working with the University of Strathclyde on some initial work to essentially target some representative lipid nanoparticles, looking at about 100 nanometers in size with very high encapsulation efficiencies, and the documented recommended PDI, which is the polydispersity index. Essentially lower is better, but 0.2 is typically the recommended level for near monodisperse there. And as you can see, for the 4 samples that we produced with the equipment, we were able to meet all of these targets and demonstrate that reproducible size control, very high encapsulation efficiency of 97%, and those low PDIs indicating that very high degree of control for the process as well, ranging from 0.19 to 0.21. We've then gone on to expand this. This is based on 500 milliliters a minute of material being produced, and we've been able to expand that initially lower down to 120, then upper to 1.2 liters a minute as well, all from one of those single AXF1 devices. Using the same formulation, and in this data showing how we can control the size of the particles that we're producing as well. Essentially what we're actually changing here is the shear that we're applying inside of it, and show that we have this scalability whilst maintaining really narrow size distributions, even decreasing polydispersity index even further as we go on. And because we're using essentially the same membranes inside each of our devices, this gives us that really good scalability across the other devices in our range. Gives you that high confidence that any initial results can easily be scaled up further if you choose to continue on and make larger and larger quantities as are predicted to be needed for these types of materials. Additionally, because we can run all of this continuously, can function very well with inline DLS measurements that you can attach in via a control loop, and that can give you that control over the size. We're demonstrating control over the size by controlling the flow rate here. So you can imagine if you're continuously monitoring that in line, you'd like your size to be smaller, you can adjust the flow rates as needed. Outside of just controlling via the total flow rate, we've also been able to demonstrate control by changing the pore size of the membrane and the size of the annulus within the device as well. Because what if you wanted to produce those 100 nanometer particles, but at 1.2 liters per minute, as we showed on the slide before? And that's where these other levers come into play. That's where being able to change your membrane pore size, being able to change the size of that annulus, lets you achieve the size you need at the flow rate you need. It decouples those two indicators. So as you can see, changing the pore size there gives us a change in the size on the left, and changing the annulus size, effectively changing the shear or the velocity within the channel inside the device. We're also able to change the size of the particles that we're producing as well. This has given us a really predictable system to allow for easy inline adjustments. As we said, we think this can really be integrated quite well into manufacturing facilities that are using things like PAT. Finally, on the smaller end of the scale, talked a lot about larger flow rates that are obviously very relevant for production, trying to get as much material produced to get as many vaccines produced as we need. But all of these things start off small on a lab bench. And that's where our most recent piece of equipment that we developed, the Axf Mini, comes in. This is our smallest device, designed to run at that 1 milliliter per minute up to 20 liters per hour potentially, which sounds like a lot, but when it's continuous with extremely low hold-up volumes, as you can see, it's just as happy producing those 1 to 2 milliliters as it is producing those 20 liters, all from one small device that you can have set up on your bench within a day. As you can see here, that same trend that we demonstrated with the AXF1 holds true, but at flow rates that are a fraction of what we were running with the AXF1. So giving us that same control over the particle size. And all of this because we're essentially using that same membrane technology with all the different devices. So the full series that we've developed for this versatile lipid nanoparticle manufacturing is our Axf series, starting from the Axf Mini at 1 milliliter per minute to 20 liters per hour. You can then move to the AXF1 for that intermediate. And just in case 200 liters per hour of material isn't enough for you, we can go to our AXF-N system, where N is the number of membrane modules that we fit inside of it. All three devices designed on the same sort of principles of let's have the lowest hold-up volume possible. Let's all use the same types of membranes, the exact same droplet formation procedures, and essentially run as simply and easily as possible to produce the materials that people need to actually develop these vaccine products. So all designed with absolutely minimal consumables, maximum ease of use, and maximum scalability as well. Thank you very much for allowing me to present a quick overview of our technology. It's absolutely fantastic to be featured in these awards alongside lots of other great companies as well. And yeah, thank you very much for the nomination, and best of luck to everybody else as well.
Okay, thank you, Sam. We'll once again pause for questions. So if you would like to get in touch and haven't already done so, please use the questions box now. And while I give you a moment on that, don't forget there are more IChemE Award webinars coming up throughout the next couple of days. You can find all the dates and times by visiting icheme.org/global awards. We have our first question. With such small holes in the membranes, what kind of SIP/CIP cycles are required to be used?
Yeah, so the membranes are generally cleaned out of place. We have worked with some of our customers on some clean in place cycles, and it does very much depend on the types of materials that they're using. Some of our other customers will use these as single-use items. So these essentially steamed out of place, assembled in an isolator, and then run that way. But yeah, as you've correctly identified, you essentially have a nice little porous substrate there that can be a little tricky for those SIP and CIP procedures. We have one that we worked on that I can't disclose too much about, but it's generally done out of place traditionally.
Okay, thank you. We're going to move on. So thank you very much, Sam. Next up is the joint entry from North Carolina State University and Photocide Protection. Please welcome Riza A. Giladi.
Wonderful, thank you. Good morning, good afternoon, and good evening to everyone who's joining us from around the world today. My name is Reza Galati. I'm the Chief Technology Officer for Photocide Protection, and on behalf of my colleagues Rich Spontak and Frank Scholl, it's a pleasure to tell you a little bit about our work on universal photodynamic coatings for infection control, for which we were nominated as a pharma finalist for the IChemE Global Awards 2021. I want to say that this work is significantly more advanced than when we were nominated back in 2019, and so we're excited to share the new results with you this morning. Just to give you a little bit of an overview of our product line, Photocide Protection is a small business. NC State University, of course, is a university, and together we are developing the Safe Light product line, which is the next generation family of environmentally friendly products that can inactivate a wide range of pathogens. It doesn't matter if they're bacteria, fungi, or viruses. And it doesn't matter if these pathogens are drug resistant or drug susceptible. And our goal is to be able to inactivate these pathogens from surfaces that lead to the transmission of them to new hosts. So we're really trying to stop the transmission of pathogenic organisms or viruses, in particularly located in hospitals, healthcare facilities, and potentially PPE manufacturers as our beachhead markets. We've received significant funding from the National Science Foundation, the Army Research Office, and we have a patent on our technology as of earlier this past year. The reason why our technology is needed is that there is really an unmet need for antimicrobial materials in the marketplace, whether that's in hospitals or aggregate living conditions such as nursing homes or daycares, gyms. All of these locations enable the transmission of pathogens, including superbugs. These superbugs arise from the overuse of antibiotics, for example, from having too much triclosan in consumer products, and they lead to drug resistance in these bacteria and other pathogens. There are other serious threats that require the need for antimicrobial materials. That includes the prepackaged food industry or the food service industry. Of course, fungi, mold, and mildew, for those of us living in damp environments, continue to pose threats to human health and disease. Bioterrorism is one where having antimicrobial PPE could be a benefit, but of course, antimicrobial PPE comes into play with what we have seen over the past couple of years with the coronavirus pandemic. Suffice it to say that the current antimicrobial products that are out there on the market really don't meet the demand for more effective, sustainable, and longer lasting protection than traditional cleaning products such as bleach. So if we take a look a little bit into more detail, particularly for a market such as hospital-acquired infections, really the numbers are staggering. So here in the US, there's about 2 million hospital-acquired infections each year. It's about 5% of all admitted patients. The cost is in the tens of billions of dollars, but much beyond the cost of dollars is the cost of lives. And so more than breast cancer, motor vehicle accidents, and AIDS combined are the number of people who die in the US each year from hospital-acquired infections. So going to the hospital for one ailment and coming down with a hospital-acquired infection while you're getting treated for that other ailment is the 6th leading cause of death in the US. The numbers in Europe are not any better, about 4 million HAIs in EU countries annually. It's about, again, 5 to 6% of admitted patients, 90,000 people each year dying. In the UK, there's about 650,000 annual HAIs. Again, 5% of all patients, translating to about 22,000 deaths, costing the NHS billions of pounds to treat. And really, this picture from the US Centers for Disease Control shows why the problem is so difficult. Each one of these surfaces represents what is supposedly a cleaned surface in a hospital environment. However, because of improper disinfection or because surfaces are easily recontaminated, pathogens can fall onto these surfaces and reside for weeks or even months, waiting in a dormant stage until they can get transmitted to a new host, usually hosts that are immunocompromised or in the hospital for some ailment are more susceptible to the transmission of these pathogens. And so the focus of Photocide Protection and NC State University as a joint entry is, can we design new materials that are inherently antimicrobial to create self-sterilizing surfaces to protect us against pathogenic bacteria, viruses, and fungi. And there are a number of existing products out there on the market, and each of these has very good efficacy. However, a lot of these products also have a number of disadvantages. So for example, traditional disinfectants, such as bleach, do not prevent recontamination. So you might spend hours cleaning a surface, but the next moment someone comes and touches that surface, now it's recontaminated with pathogens. Often they're only effective against one class of pathogen. Their germicidal activity will be consumed as a function of time. There could be the emergence of drug resistance to many of the antimicrobials included in those products. But either the leaching of the biocide or the use of environmentally hazardous agents is increasing the amount of regulation that is there, that is driving the need for new environmentally friendly antimicrobial materials. And so what we have come up with as an idea is using an alternative method for preventing bacterial infections, preventing the transmission of pathogens from surfaces to new hosts. And that's to repurpose photodynamic therapy, which is a US Food and Drug Administration FDA approved treatment for certain types of cancers, age-related macular degeneration, acne, and psoriasis. And it's under the review for the treatment of bacterial infections, but we're trying to use photodynamic therapy as a preventative measure to prevent the transmission of pathogens from the surface. As you can see from these images, photodynamic therapy, as the name implies, requires the use of light, visible light, to function, at least in our hands. So our solution is to come up with photodynamic coatings, and we have formulations that produce reactive oxygen species. One in particular is called singlet oxygen, upon exposure to visible light. So we take a compound, which is our drug, called a photosensitizer. That compound is a simple dye that absorbs light from any light source, visible light in our case, but it could be from an LED light, a fluorescent bulb, daylight, sunlight is fine as well, but any visible light source, and transfers that energy to molecular oxygen that's in the air, creating an active form of molecular oxygen called singlet oxygen. That's biocidal. It's a powerful diffusing bleaching agent with very strong microbiocidal efficacy. It's simultaneously effective against bacteria, viruses, and fungi, but because it operates in a similar fashion as bleach, the non-specific targeting means that it's going to be equally effective against drug susceptible and drug resistant strains. Because it's non-specific, that also means that no resistance will arise to these reactive oxygen species, such as singlet oxygen, and that our formulations are effective for up to 30 days. What's important is that these compounds are harmless and non-toxic to the environment, they're heavy metal free, and they don't harm the user. The production of these reactive oxygen species is right at the surface of the materials, and they are unable to diffuse a significant distance away from those materials, again, because they have a very short half-life. They reform breathable oxygen in less than a millisecond. And so whether it's against any of the pathogens that you see here on the bottom left of the screen, the way that our material works is that we can have a coating, as an example, that we apply to a fabric. We shine light onto that coated fabric. The molecular oxygen is activated to the biocidal form, leading to the inactivation of those pathogens. The inactivation of those pathogens can actually be seen here on these SEM images on some coated nanofibers that contain our photodynamic antimicrobial coating. Here on the left-hand side, you can see that prior to the addition of visible light, these images were obtained by SEM for materials that were kept in the dark. You can see Staphylococcus aureus as a gram-positive bacterium and E. coli as a gram-negative bacterium exhibit healthy cell morphologies. But after the addition of visible light, you can see that the generation of reactive oxygen species, such as singlet oxygen, causes wholesale oxidative damage, extensive oxidative damage, leading to the inactivation of the bacteria. They're unable to proliferate when they are in this damaged state. If they're unable to proliferate, they're unable to be transmitted to new hosts and cause subsequent disease. Now, there are a number of previously studied photodynamic materials that we and others have looked at that you can see in the literature, whether they're photosensitizers that are conjugated to cellulose fibers or grafted onto the surface of fabrics, or incorporated into synthetic nanofibers from electrospinning, or included in the bulk incorporation of an olefinic block copolymer here on the right-hand side. Whether they're surface grafted or bulk incorporation, those methods use significantly more amounts of photosensitizer, which leads to added cost. Their syntheses and productions tend to be very tedious in many cases, and thus therefore not scalable for the use that's needed to be deployed in hospitals, where you might need hundreds or thousands of square meters of materials per hospital per day. So we need an approach to overcome these limitations, and the approach that we have adopted is the use of universal photodynamic coatings. And so to introduce the coatings themselves, we can take a common photosensitizer commercially available and mix it into a bicomponent formulation with a photocross-linkable polymer, this polyvinyl alcohol polymer, SBQ-PVA, in the presence of a UV cure, creates this cross-linked web that you can see here on these SEM images on the right-hand side. So what we did was we took the bicomponent mixture, placed it in a simple airbrush, but you could do this with dip coating, kiss rolls, any type of coating technology, slot die coating that you'd be interested in using on an industrial scale. And we took from the NC State Nonwovens Institute nylon PA6 nanofibers that were made in the same fashion as polypropylene nanofibers would be made for use in N95 masks, as an example. And so we took those nylon nanofibers, but we can do this with wood or plastic or any other substrate, and we spray coated them with our bicomponent mixture, subjected them to a UV cure. And again, whether it was dip coated in the top or spray coated at the bottom, you can see that we've created this very nice web of our photocross-linkable polymer, into which has been embedded the photodynamic agent, which is the drug that is going to be inactivating the pathogens. And so when we take these materials, whether they're dip coated or spray coated, and we do some antibacterial studies to determine their efficacy, as you can see here on the left-hand panel, against methicillin susceptible Staphylococcus aureus or against antibiotic resistant E. coli, again, gram-positive and gram-negative pathogens respectively, we have fairly good inactivation to our minimum detection limit, 100% inactivation of the Staphylococcus aureus, and 3 log units, 99.9% inactivation of our antibiotic resistant E. coli. Again, the y-axis here for these panels is always going to be shown in a logarithmic scale. If instead of looking at gram-positive and gram-negative pathogens, bacteria, we look at viruses, we can look at human coronavirus strain 229E. This is a surrogate of the, this is a common cold coronavirus that is a surrogate of the SARS-CoV-2 coronavirus that's responsible for the COVID-19 pandemic. But it's at a biosafety level that we can accommodate within our laboratories at the company. You can see that in the photosensitizer free, when we don't have the drug present, these materials are unable to inactivate any of the virus. However, in the presence of our photosensitizer embedded within the coating, we're able to inactivate coronavirus depending on the intensity of the visible light that's being used. Anywhere from 99.7 to essentially our minimum detection limit, 100% inactivation. And again, this is conferring the antimicrobial properties through the use of our universal photodynamic spray coating to nanofibers. However, we can also do that to medical textiles and upholsteries. So we were gifted from another company, Vescom America, a number of different medical textiles and upholsteries that have the names Husk, PRU, Capri, Deans, and Woolen, located here on the left-hand side of the photographic image. Because our compounds, such as this zinc porphyrin containing photosensitizer, are visible light absorbers, they do have a slight color associated with them. So you can see in the photographic images on the left, a little bit of a color change in the materials, and that can be accounted for in the dyeing processes of the original materials, should that be an issue for companies. In the SEM images, you can see the uncoated fibers versus the spray-coated fibers, again, using a spray coating that's a bicomponent mixture of our photodynamic drug and our photocross-linkable polymer. And based on those images, what we have been able to establish is that our coating thickness is relatively thin compared to the fibers. It's only about a micron or so. And that leads to a very economical use of the photosensitizer. So photosensitizers can be costly if they're incorporated into the bulk, but when they're only applied as a thin coating, they actually are relatively inexpensive. In fact, only a fraction of the cost of the materials themselves, the upholsteries or the medical textiles themselves. So this is a very economical approach. Again, Staphylococcus aureus, these medical textiles, depending on the medical textile itself, it's always the same coating, but how much the coating was successfully sprayed onto the material, we get anywhere from 97 to 99.999% inactivation of the gram-positive Staphylococcus aureus, anywhere from 92 to, again, 99.999+% inactivation of human coronavirus, again, using this porphyrin-based photosensitizer. We can, instead of using that more unique photosensitizer, use something that's very common, that's registered here in the US with the Environmental Protection Agency, such as methylene blue or rose bengal. Again, two very common commercially available photosensitizers that have other applications in the medical community as well. We can incorporate either of these three photosensitizers into our bicomponent spray mixture and evaluate their efficacy against Staphylococcus aureus, for example, human coronavirus, feline calicivirus, which is an example of a small non-enveloped virus, which is a surrogate for norovirus, or human adenovirus, which is a large non-enveloped virus. And in all cases, we're able, depending on the photosensitizer, to get up to 99.99% inactivation of these various different pathogens from the surface of a medical textile or upholstery. And so with that, just to summarize, the work that I presented has been in two publications. The initial publication here is the 2021 publication in ACS Applied Materials and Interfaces. The medical textiles work was just recently published in Frontiers in Medicine, also this past year. And so we're able to make these antimicrobial photodynamic materials through a variety of high throughput methods. As a company, we're particularly excited about the spray and dip coating methods because these are translatable to manufacturing and easily scalable. We've been able to extend this work to include polyacrylonitrile, nylon, block copolymers, and again, medical textiles and upholsteries, to show that we have very good to excellent antibacterial activity against a whole host of gram-positive and gram-negative bacteria, whether they're drug susceptible or drug resistant, and also very good to excellent antiviral activity against all classes of viruses, whether they're envelope viruses or non-envelope small or large variants. And again, we're very excited to be placing these into hospitals, healthcare facilities, and PPE manufacturers because our coating has our coatings have a number of different advantages. Again, they're very economical. They use very little photosensitizer, and as such, we can utilize inexpensive and commercially available photosensitizers that will enable us to scale to the sizes needed for deploying this throughout the various different medical markets. They're eco-friendly. We use a waterborne polymer, so we don't have any VOCs, a facile curing process, and we're able to use a capping layer if we want, so we can prevent photosensitizer from leaching into the environment from the disposal process. They have some very favorable properties. They're tough, abrasion resistant, they're optically transparent. They're easily integrated into manufacturing processes, again, through spray or slot die coating or kiss roll coating. They can be used on a wide variety of different substrates or backings, and they're highly and broadly antimicrobial. And so we are developing now our product line with our final formulations being safe tape and safe film for covering high-touch surfaces, and our spray that we can integrate into PPE production. Again, going after the medical markets initially, but a whole bunch of different or a wide variety of different consumer markets are available to us, given that this is a platform technology. And so with that, just wanted to conclude by saying we are honored to be selected as a finalist for the 2021 IChemE Global Awards competition. Thank you for your attention, and if you would like to reach out to us, contact information for us at NC State University or Photosite Protection is shown here on the slide. So once again, thank you for your attention.
Okay, thank you very much, Reza. We're going to move straight on to our final presentation. We are running a little bit behind time. So this is the joint entry from the University of Sheffield, Perceptive Engineering and Siemens. Once again, as a pre-recorded presentation, but with live Q&A to follow. So let's hand over to Mo Zandi for the presentation.
Hello, I am Professor Mo Zandi from the University of Sheffield. This is a double act presentation, and alongside my colleague, Dr. Eduardo Lopez Montero, Senior Engineering Consultant at Perceptive Engineering, is helping me to share the project results with you. The University of Sheffield installed a world-leading pilot plant within the Faculty of Engineering in 2018. It is named the Diamond Integrated Pilot Plant, or for short, DEEP. DEEP is capable of manufacturing pharmaceutical tablets from blends of model active ingredients and inactive media. This includes key powder process steps for formulated products manufactured, such as crystallization, blending, granulation, and tableting. The new continuous powder processing plant emphasizes the importance of complex particulate products and formulated products more broadly in modern chemical engineering. And we are also reflecting this in our new curriculum in chemical engineering. It is truly research-led teaching. DEEP's key features include a dedicated industrial control room to accommodate an advanced control system and a digital twin of the plant. The continuous powder processing plant in DEEP, including the continuous crystallizer, were provided by different manufacturers and vendors, and also were not IoT enabled. University of Sheffield, Perceptive Engineering, and Siemens joined forces to develop an innovative solution to digitalize the whole continuous drug manufacturing process from crystallization to tableting machine, and converted into the world's leading Industry 4.0 demonstrator. The main objective of the project was to adopt a data-centric approach and automation, and to demonstrate facilities to test and validate potential IoT applications in the real world.
In this slide, we list the challenges and goals of the project. The challenges or previous state of the pilot plant included a low digital process maturity. In other words, the pilot plant was not IoT enabled. Each system and process unit represented its own isolated data island. Therefore, data had to be locally saved on separate locations and then brought together manually for offline analysis. Also, existing knowledge from technical personnel could not be fully leveraged when controlling the processes, ultimately making the operation of the pilot plant highly manual, resulting in a considerable workload on operators. Therefore, the University of Sheffield wanted to develop a solution that could improve the system operation and overcome these challenges. The main goals of the project were to marshal and align all data from the different systems in a centralized location, develop dashboards for monitoring the systems locally and remotely, a solution that could use all the data in real time and is able to use machine learning techniques to model, predict, and ultimately control critical process variables. Also, to implement a solution that can upload in real time all data to Siemens MindSphere, so the data can be used across the university for data science research activities. The first task for the development of the control system for the pilot plan was to set up the digital architecture. This architecture can be summarized in this block diagram. Essentially, Perceptive's PharmaMV software was used to read and write data to and from all sensors and process units, including Siemens MindSphere, and the software used to run the digital twin. This facilitated access to all data from a single interface. This digital architecture enables a synergy between statistical and nonlinear models, providing a complete set of tools for monitoring, advanced process control, and machine learning, a seamless integration with process units and PIT instruments, and also enables data fusion and alignment across the full platform. After PharmaMV was set up as the single-point repository for all equipment's data, the optimized experimental design platform was configured for each of the process units at the Diamond pilot plant. The OEDP is a solution built upon Perceptive Engineering's PharmaMV software and provides the workflows and tools for rapid process development using optimized experimental design techniques. These techniques include automated design of experiments, machine learning for self-optimization and automated model adaptation, response testing, and integration with mechanistic digital twins for development and deployment of advanced process control. The OEDP includes pre-configured tools and embedded documentation to reduce overall configuration and development effort. All these tools can be accessed via interactive dashboards, so operators at the University of Sheffield can navigate across all the process units, drill down to specific parts of the system, and activate any of the digital tools using such interactive dashboards. The platform also supports user authentication with different levels of access. For example, users that are only allowed to monitor the system to administrators that can trigger APC and make changes to the OEDP solution. In summary, the OEDP enables smart process development for the crystallizer, filter dryer, and the continuous tableting line. The current slide shows how a model can be identified to predict the torque of the twin screw wet granulator. Statistically rich data for modeling torque dynamics can be generated using the OEDP's response test tool. Then, through machine learning techniques, the platform is able to identify the model automatically and also to adapt the model to new operating conditions if needed. An example of an automated step test is displayed in the right-hand side of the dashboard. Real-time torque measurements are displayed in green on the top trend. The other trends show in red the steps applied by PharmaMV and in blue the readbacks from the process variables. These data are used in real time to adapt the model, and after a predefined time has elapsed or a model accuracy level is reached, the automated modeling stops and the new model is integrated within an MPC architecture to control torque to set point. Operators can activate and configure the automated model adaptation via the panel at the left-hand side of the dashboard. The platform will continuously monitor the quality of the data and will notify operators of any interface communication errors. The platform will stop any response test or DOE if such communication errors are detected. It is worth to mention that the data from any automated response test is uploaded to MindSphere in real time to allow scientists across the University of Sheffield access to the data for analysis and research activities. One of the main goals of the project was to upload the data in real time to MindSphere, which is Siemens' leading industrial IoT as a service solution. 2 Siemens nanoboxes were interfaced to PharmaMV to enable the possibility of pushing data from all process units to the University of Sheffield's MindSphere account. The screenshot in this slide shows how data from the crystallizer unit operation is collected by PharmaMV and then uploaded into the cloud in real time. The data can then be accessed both online and offline by students and scientists at the University of Sheffield.
The importance of big data visualization as a tool for Industry 4.0 is widespread and well documented. An example of the benefits of this project is the integration of web-based data visualization tools such as Wiz through MindSphere to give both industry and academia access to the data. Here, Wiz provides a one-stop shop for data analysis and visualizations. Scientists and non-scientists can use the platform to better understand the data, analyze it, and make informed decisions.
This is a summary of the benefits of the digital pharmaceutical manufacturing solution developed for the Diamond pilot plant. The digital control system allows to adopt a data-centric approach and automation, making knowledge about formulations, process and manufacturing of pharmaceuticals accessible and reusable. Enables a cost-effective experimentation with production-class operational equipment without the costly downtime and reduced output if that were to be done on actual production machines. Provides an opportunity to access and optimize low-level control systems in a pre-competitive environment, and allows development of a service-oriented business model. The digital control system was developed on Perceptive's PharmaMV software, which provides a full suite of analytical modeling and APC tools for rapid process development and optimization. It allows fusion and visualization of process and analytical data in real time. For the pilot plant, PharmaMV enables the prediction of critical quality attributes using data-driven models and digital twins. It also enables the implementation of model predictive control for CQAs and can be interfaced to Siemens MindSphere to transfer data in real time to the cloud. Some of the main benefits of this digital control system are that it provides the university with a demonstrator of a state-of-the-art automation using an industry-proven 21 CFR Part 11 compliant platform, delivering a practical learning experience of an industrial continuous powder processing plant to undergraduates and access to manufacturing plant equipment to research teams. It provides automatic data fusion and visualization using multivariate trends, charts, and interactive dashboards, automated execution of DOEs, process response testing, which reduces operators' workload and streamlines data generation for optimization, analysis, and research. Online model adaptation using machine learning techniques allows the evaluation and deployment of process condition monitoring, APC, optimization, and soft sensing approaches, and enables cloud connectivity of all process units to support research and development across all data science activities.
This project was a team effort, and I would like to acknowledge and thank all the colleagues who were involved and supported the project. Without them, the project couldn't happen. I would like to thank all the colleagues from Perceptive Engineering, University of Sheffield, and Siemens. Also, I would like to thank Eduardo, and thank you for your time and giving us this opportunity. Thank you.
Okay, that was a pre-recorded presentation, but Mozandi and Eduardo Lopez Montero are with us. So we'll pause for just a moment, and if you have any questions, now is the time to get in touch. And if you have been suitably inspired by today's presentations, it might be time to start thinking about your own awards entry for next year. Keep an eye on the IChemE website for more details, but we plan to open for entries in early March 2022. Okay, we don't appear to have any questions, so thank you, Mo and Eduardo, for joining us. We're going to move on, and we've reached the moment we've all been waiting for. It's time to find out the winner of the IChemE Pharma Award, sponsored by PM Group. Before we do that, I'd like to thank all our finalists for joining us today and being with us. I'm sure you'll agree a fine range of projects. So as you can see, our highly commended entries are those from GSK and the joint entry from North Carolina State University and Photosite Protection. Well done to everyone associated with those projects. But now to announce the award winner, we'll hand back over to PM Group's Peter Farrelly.
Thank you. Some very interesting presentations, and everyone I think would be worthy of winning. But ladies and gentlemen, the winner of the IChemE Pharma Award, sponsored by PM Group, is Micropore Technologies.
Okay, congratulations to Micropore Technologies then, and to everybody who worked on that winning entry. Sam, if we can get you back with us, if you are still there, tell us, how does it feel to be an IChemE Award winner?
That's excellent. Excellent. Yeah, thank you very much, and congratulations to everybody else as well. Lots of really great research done out there, and it's great to see everybody having the chance for recognition at these sort of events. But this is going to mean a huge amount to all the team. I'm sure they'll be ecstatic when I tell them, albeit virtually at the moment. But Yeah, thank you very much. Real, like I said before, honoured to be featured alongside such other prestigious institutions and organisations as well. Yeah, thank you.
Congratulations once again, and congratulations to the whole team, as you say, Sam. So well done to everybody. Thank you to all of our finalists, and thank you once again to our sponsor, PM Group. We've still got plenty more IChemE Award webinars to come over the next few days. We're back tomorrow to announce the winners of two more awards, but that is all for now. So good afternoon, and thank you for joining us.